Neuroscience just did the impossible. Something monumental just happened in neuroscience, a breakthrough that pushes past the known boundaries of what we thought was possible. For decades, we've been able to see the brain light up with activity, but we've been watching from a distance, like seeing a city glow at night without knowing what's happening on its streets. We could see the traffic, but not the roads themselves. Now that has changed forever. In a move that sounds like it was pulled from the pages of a science fiction novel, researchers took the deadliest virus on the planet, systematically disarmed it, and rewired it. They transformed this lethal pathogen into a microscopic lantern and used it to illuminate one of the greatest mysteries of the human mind: how a single dose of psilocybin can physically repair the brain. They watched it happen in real time, all the way down to the individual synapses, the exact neurons involved in this profound transformation. What they discovered is a paradigm shift. It fundamentally changes our understanding of consciousness, the nature of personal identity, the deep-seated roots of trauma, and the very essence of who you are as a human being. If you have ever wondered how one psychedelic experience can completely change a person's life, how it can lift the weight of lifelong depression, dissolve the grip of addiction, or reframe the trauma of the past, you are about to see the actual physical mechanism behind it. And I need to be crystal clear. I do not say this as a metaphor. This is not a philosophical discussion or a vague reference to neuroplasticity as a popular buzzword. I am talking about a tangible, observable process of programmable rewiring, captured on camera with a tool that was, until now, a legitimately weaponized virus. The implications of this are staggering, reaching into medicine, psychology, and even our core definition of self. We are on the cusp of understanding the brain not just as a fixed biological machine, but as a dynamic, editable architecture. Stay with me. Because the true breakthrough is not just what psilocybin does to the brain. It is the revolutionary fact that we can now physically map, trace, and ultimately control what it does. We are moving from hoping for a positive outcome to engineering one. The final part of this video will reveal a level of precision and potential that may be the most astonishing thing you have ever heard about the human mind. It suggests a future where we can guide the healing process with surgical accuracy. The part that feels like science fiction. To understand the magnitude of this leap, we have to go back to 2012. That's when the scientific community first confirmed that psilocybin, the active compound in magic mushrooms, literally grows new connections between neurons. Not just a few, thousands of them, millions of them. These new dendritic spines, the physical branches that form new synapses, sprout with astonishing speed. Some of these new connections last for weeks. Some appear to be permanent, laying down new hardware in the brain. But this discovery, as profound as it was, came with a major problem: a black box. We could see the growth, but we had no idea where those new connections actually went, or what they were doing. We knew the brain was furiously building new roads, but we had no map. We had no idea which cities those roads were connecting. Were they building helpful new highways between regions that needed to communicate, like sensation and memory? Or were they creating confusing back alleys and dead ends? Without knowing the destination, the therapeutic potential of psilocybin remained a brilliant but blurry promise. This is where a collaboration between Cornell University and the Allen Institute for Brain Science did something almost unthinkable. Faced with the impossible task of tracing billions of microscopic connections in a living brain, they turned to a tool of terrifying efficiency. They turned to rabies. Yes, you heard that correctly. The virus that causes one of the most feared and fatal diseases known to humanity. The virus that makes animals foam at the mouth. The reason rabies was chosen is because of its uniquely insidious method of attack. The rabies virus naturally travels backward through the nervous system. It's a retrograde invader. When it infects a single neuron, it doesn't just stay there. It cleverly jumps backward across the synapse into every single neuron that sends signals that neuron. Then it repeats the process, jumping upstream again and again, lighting up an entire input chain. From a virology standpoint, it's a nightmare. But from a neuroscience standpoint, it's the ultimate retrograde neural tracing system. It's a biological tool that can perfectly map every connection leading to a single point. There was just one small problem. It is the deadliest pathogen we know of, with a nearly 100% fatality rate once symptoms appear. Using it for research was out of the question, until now. In a stunning feat of genetic engineering, scientists created a new version of rabies. First, they deleted the gene that allows the virus to replicate and spread uncontrollably, rendering it non-lethal. They effectively took the bullets out of the gun. Next, they modified its coat protein so that it could only jump backward across one single synapse, and then stop. It could no longer cascade through the entire nervous system. It was a single-step tracer. Finally, they inserted a gene from a jellyfish, the gene that produces green fluorescent protein. This turned the virus into a biological beacon. They took the most deadly virus in the world, stripped it of its lethality, and turned it into a controllable, trackable, biological neon green highlighter, a tool that could safely and precisely illuminate the brain's hidden wiring. With this incredible tool in hand, they could finally answer the question. They administered psilocybin to their animal models, and then, using this modified virus, they could target specific neurons and see exactly where the new connections were coming from. A virus that was built to kill is now being used to illuminate the very circuitry of healing inside the nervous system. And what it showed them will change the future of mental health forever. The study on day 1, researchers injected a helper virus into the frontal cortex of mice to prime specific neurons. That incubated for 13 days. On day 14, one group received psilocybin, the other received saline. On day 16, the modified rabies virus with the green marker was injected into the same brain region. On day 21, the virus had spread backward exactly one synapse. Then the brains were harvested. Using light sheet microscopy, researchers photographed millions of unimaginably thin slices of each brain. They counted every green neuron. Over 500,000 inputs per brain were scanned and mapped across 316 brain regions. This was the first complete map of how psilocybin rewires a brain ever created in history. What they found. The results were expected to be random. They were not random at all. The rewiring followed a pattern so statistically precise that the probability of it happening by chance was listed in the study as p equals 0.006. Something very specific was happening. When the researchers analyzed the brain-wide connectivity maps, two distinct and opposing patterns emerged with stunning clarity. The first was a significant strengthening of connections, an increase of up to 10%. What was remarkable was where this strengthening occurred. It wasn't random. The changes were concentrated exclusively in the brain's sensory and motor regions, the very circuits that process the outside world. We saw it in the primary somatosensory cortex, the brain's hub for touch, temperature, and the physical sensation of your body in space. We saw it in the primary visual cortex, which constructs our reality from light and shadow. The motor cortex, responsible for planning and executing movement, also showed this increased connectivity. And critically, so did the retrosplenial cortex. This region acts as our internal GPS, governing spatial memory and our fundamental sense of where we are in the physical world. It's the part of the brain that helps you navigate a familiar room in the dark. In essence, psilocybin was systematically reinforcing the brain's connection to the external physical world. The subjective reports from psychedelic experiences of colors appearing more vibrant, sounds having more texture, and a profound sense of being present in one's environment suddenly had a clear neurological correlate. The world feels more vivid on psilocybin because your brain is literally, physically, rewiring itself back into that world. It's not just a feeling, it's a biological event. The brain is dialing up the gain on reality, prioritizing the raw data streaming in from the senses over its own internal chatter. But this was only half of the story. For every connection that grew stronger, another set of connections was doing the exact opposite. The second pattern was a profound weakening of connections, a reduction of a full 15%. And just like the strengthening, this wasn't random. These changes were all located in a network of regions responsible for constructing our internal narrative of who we are. This network is where the self lives. The infralimbic area, which handles learned fear responses and emotional regulation, went quiet. The insula, a key node for interoception, our sense of the body's internal state, and a vigilant detector of anxiety and threat, also saw its connections diminish. The hippocampus, the great archivist of memory that provides the backstory for our identity, loosened its grip. The amygdala, our primal emotional processing center, became less influential. And finally, the orbitofrontal cortex, the part of the brain where obsessive thoughts, rumination, and rigid expectations are forged, showed a significant drop in connectivity. In plain language, psilocybin temporarily disassembles the very scaffolding of the ego. It quiets the brain regions that generate your sense of self, your ingrained fears, your looping memories, your self-referential stories. This collection of regions is famously known as the default mode network, or DMN. In many mental health conditions, particularly depression and anxiety, the DMN is overactive. It becomes an echo chamber of negative self-talk and rigid, pessimistic predictions. Psilocybin doesn't just turn down the volume on this network, it fundamentally loosens its structure, it loses its grip. This dual action, turning up the volume on the outside world while turning down the volume on the internal self, is the key. It explains why people so often describe these experiences as transformative, feeling like they are seeing the world for the very first time. Because for their brains, stripped of the usual filters of memory, fear, and self-consciousness, they are. Neurologically, the world knew. But the study revealed one final crucial detail, a finding that points toward the future of this research. The scientists called it the part nobody expected. In one phase of the experiment, they decided to chemically silence just one specific brain region during the psilocybin session, making it unable to communicate with the rest of the brain. The result was profound. That single silenced region did not get rewired. It was left untouched by the psilocybin-induced plasticity. Every other active region in the brain, however, rewired itself exactly as predicted. This demonstrated a critical principle. For the brain to be changed by psilocybin, it must be active and engaged during the experience. The rewiring isn't a passive chemical reaction, it's an activity-dependent process. The brain rewires what it uses. The rewiring is not random, it is context-dependent, it is experience-dependent, and it is programmable. The study is essentially proving that when your brain is growing, you become whatever you pay attention to. That means if you know which pathways will be active, you can choose which ones get strengthened. If you can quiet the circuits driving fear, rumination, and trauma during that window, you can weaken them significantly. If you want to strengthen visual processing, show visual stimuli during the session. If you want to reduce anxiety loops, quiet the insula or the amygdala at peak plasticity. If you want to reshape someone's entire sense of identity, you guide their attention into new self-models while the old ones are temporarily offline. What this actually means. We are crossing a threshold in neuroscience that may be the largest in history. For the first time, we are not just watching a brain change, we can now watch what a brain is becoming, and we can influence that process with precision. This is the first time science has identified the exact circuits that shift during a transformative experience. We are seeing the real neurological blueprint behind every story of someone healing from PTSD in a single session, or being fundamentally changed in one night. And we are seeing it neuron by neuron. Once you can see the circuitry, you can design it. And here is where this becomes a double-edged discovery. Identity, the thing you call yourself, is not a constant. It is a product of which networks are active. And those networks can now be turned up or turned down. Your personality, your memories, your fears, your reactions. They are not as permanent as they feel. That truth is liberating. It gives us a mechanism, a tangible process for intentional self-transformation. But it is also deeply unsettling, because it means that during this period of profound neuroplasticity, our sense of self is exquisitely vulnerable. Whoever or whatever guides your attention during that psychedelic window gains a level of influence that is unprecedented. They are not just shaping an experience, they are helping to select which neural pathways get reinforced and which get pruned. They are, in a very real sense, co-authoring the version of you that walks out on the other side. This isn't about simple persuasion. It's about directing the biological process of belief and identity formation itself. This discovery carries the immense power to heal millions. Imagine a veteran with PTSD, whose attention is gently guided away from traumatic memories and towards feelings of safety and connection, effectively weakening the neural circuits of fear. Or someone with severe depression, whose focus is shifted from patterns of self-criticism to a renewed appreciation for the world, loosening the grip of the brain's default mode network. But this exact same mechanism carries the power to manipulate. In the wrong hands, a coercive group, a bad actor, or even a commercial entity with a profit motive, attention could be directed to instill specific beliefs, create artificial loyalties, or even erase inconvenient memories. The potential for misuse is profound, and it demands we proceed with extreme caution. The bigger picture. What this study reveals goes far beyond therapy or trauma treatment. It forces us to confront a revolutionary idea, that consciousness may not be a fixed, immutable property at all. Instead, it might be a dynamic, programmable operating system. For the first time, we can see what looks like the underlying code running the software of human experience. And once you see the architecture, you cannot unsee it. Every thought, every feeling, every perception starts to look less like a mysterious, emergent property and more like the output of a biological program, a program that can, under certain conditions, be edited. This insight reframes everything. But to truly grasp its implications, you need to understand the next layer of the puzzle: the brain's prediction engine. Why does the brain loosen its grip on its own model of reality during these states? How does this predictive model shape every single moment of your waking life, from the colors you see to the emotions you feel? The next videos we are releasing this week and next go deeper into this predictive processing framework. Once you understand how your brain is constantly guessing what's going to happen next, and how psychedelics disrupt that process, everything about this discovery gets even more fascinating and, frankly, more bizarre. This is a landmark moment for neuroscience, a turning point in our understanding of the mind, and I am glad you were here for it. If you want to continue this journey with us, make sure to like and subscribe. I'll see you in the next one. Thanks for watching.